Goodman, a clinical professor here in the Department of Medicine, in the Department of Cardiology at the Leon Charney Division of Cardiology. And welcome to our lecture series. I want to say thank you so much, those of you who came out. Anya and I went back and forth for the last 3 days, every few hours, as to whether or not to cancel tonight. Literally, we were calling all the different weather stations to try to get. And in the end, we decided that if there's one person who shows up, because they didn't hear that we canceled it, we were not going to let them down. And that's our commitment to you. And I'm really grateful to those of you who came out. And I'm especially grateful to Anya, who makes this happen. I thank her every time. Really appreciate all she does to make this program so successful. And I'm going to introduce our speaker in a second. I just want to tell you that we are still going to have lectures in January, February, March. Stay tuned. You're all on our email. If not, please make sure that you give it to Anya. We're going to talk about stroke and heart disease again next year. And we're going to, in February, it's Heart Month. We're probably going to give you a whole update on what we do in our Cardiac Prevention Center here and what kind of research we're doing. It's really exciting. And then I decided that climate change, it's crazy. There are people who actually believe it's not happening. And there are people who say it absolutely is. So we thought we'd get an expert on that and have a discussion about, is it real and how does it affect our health, particularly our cardiovascular health? And of course, you don't have to be too much of a scientist to know that storms and all sorts of natural disasters cause severe stress. And there are many, many, many people who've died of heart attacks, never mind the flood and everything else, just because of the stress of things that happen because of climate change. So stay tuned for that. Here's some information about our program. Here's our website, www.nyulangone.org/heart health lectures. You can join our email list, as I told you, and the email is hearthealth@nyulangone.org. I'm saying some of this because we're recording this, and there are people who are going to be listening to this lecture because they couldn't make it in. So that's that. Many of the past topics, I put them up. It's quite, I'm very proud of this. These are, and this is partly for our speaker to see all the different kind of topics and things that we've spoken about. This is all about prevention and education and empowering people to learn about what they should do and what they can do to be healthy and to prevent events first time, second time, third time. So we go out of our way to bring topics to people that are related to cardiovascular health. So tonight, it's my pleasure to introduce Dr. John Pappas, who is an associate professor here in the Department of Pediatrics. He is a wonderful man, and I called him last night and I said, I don't know how many people are going to show up. And he said, don't worry about it. That's what kind of human being he says, if there's one person, I'm there. So thank you so much for coming. He is somebody that is an absolute expert in genetics, and I chose this topic because I'm fascinated by it as well. I think it is so important now because we're getting so much information now, which you're going to hear about. And my question to him, and what I myself like to talk about, is should you know your own genetics? Does it help to do this testing? Is it going to make a difference? Are we ready to take this to prime time with our patients? So a little bit about Dr. Pappas. He did his fellowship at Beth Israel Medical Center and his residency in Beth Israel Medical Center in Pediatrics, and he got his medical school education at the University of Athens from Greece. So he's been interested in genetics since he was an undergraduate student. He loved the idea that a genetic code is basically the same for bacteria and for humans. He went to medical school in the University of Athens, as I told you. After medical school, he did a pediatric residency. In between his residency and fellowship, he spent 2 years in the Greek Navy. So if you have any questions about that, if you're interested in maritime stuff, he's an expert there too. He started his career as an attending clinical geneticist at Beth Israel in 2001, and he's currently the director of the clinical genetic services at NYU in pediatrics. And he is a real expert on genetic syndromes. So you can imagine how busy he is with young parents who constantly worrying about, is my child going to be okay? And we have the ability now to do so much testing that we can help them predict what the child is likely, if the child's likely to be healthy, and more importantly, whether or not you should even have children if you have certain genetic information or genetic coding that would put your child at risk. So, Dr. Pappas, it's a great pleasure. Thank you so much for coming.
All right, thank you very much for coming out with the weather. So we'll discuss about the genetics and if knowing the genetics right from you from the perspective of genetic testing, not so much from family history, because you can analyze your own family history and you can learn a lot of information. But the advent of all the new technology is about genetic testing, so we'll go with that tonight. So the objective is to recognize the value of genetic testing and the limitations, and hopefully we'll have some time to delve a little bit into adverse effects and to the ethical dilemmas that come out from these new technologies. So we'll go a little bit about our genome and about our genes, what is there, where they are, and what's the structure, in order to introduce how the genetic testing is happening in the lab. And then we'll go to the clinic. I have a couple of cases to see how we practically apply that to the clinic, and we'll end with future directions and the conclusion. Is it right to know your genetics or not? So the cells in the genome, the average human body has these 37 trillion cells, and every cell, almost every cell, not every cell, but the majority, by far the majority, has a nucleus that has this chemical, the DNA, that has all the genetic information. So our genetic information is coded by chemicals in this DNA. So the DNA is a polymer, meaning that it's made from individual chemicals that have a name, the A, T, C, and G, the adenine, thymine, cytosine, and guanine. And these chemicals, actually every triplet makes a code that's particular of making the proteins, and the proteins is what directs the function and the structure of our body. So our body is made out of cells, is made out also material between the cells and material in our bones and connective tissue, but everything actually is encrypted in this chemical, the DNA that's in the nucleus of the cell. So you probably, I heard about RNA. RNA is the messenger that takes the information outside the nucleus to the rest of the cell, the cytoplasm, in order to make the protein. What's a gene? A gene is the smallest part of our inheritance, okay? The smallest part that's associated with a particular trait. And it's a unique piece of DNA. It's a DNA that is not seen in copies, because a lot of our DNA, the majority of DNA, is not actually genes. 98% of our DNA is sequences that is not genes. What's a gene? It's a unique sequence that's usually associated with RNA and protein that has a function. All right? Now, there are 20,000 genes in our genome approximately. I mean, if this question was posed 15 years ago, they would say 60,000 or 100,000. Now, after the Genome Project, we know it's about 20,000. And less than 10%, and this all 20,000 genes is less than 2% of our genome. We don't know the function of all these genes. We know actually about a third, less than half for sure. And if you compare the genomes is all the DNA that we have of two individuals. Two unrelated individuals are 99.9% similar. But this 0.1% is 3 million base pairs. So it's important. It's not insignificant because of the vastness of the genome. So I have a little video here that was very old, but it summarizes what I told you about the genome. It's going to give you a good base of biology to think why genes play a role and why it's so important, this blueprint of life. So let's try to play this.
Life, a sunny day, a simple friend, and a complex biological story. The Human Genome Project is a way of exploring our molecular cells. Almost all of our cells, the muscle cells that let us smile, the brain cells that perceive the humor in things, the cells of our eyes that take it all in, contain a complete set of all our genes, the genome. If we could journey inside ourselves into a cell, we would see 23 pairs of chromosomes packed into a nucleus. Each chromosome contains a long coil of DNA. If all the chromosomes were unwound, the DNA in just one of our cells would stretch 6 feet long. The DNA double helix contains 4 kinds of building blocks. An A always pairs with a T, a C with a G. DNA contains information to make every part of our bodies with its 4-letter language. Each of our thousands of genes codes for a specific part. RNA polymerase copies the information in a gene into a messenger molecule, messenger RNA. The building blocks of messenger RNA and DNA are called bases. The bases on one strand of the DNA specify the order of bases on the new strand of messenger RNA. The DNA always stays inside the nucleus, but messenger RNA travels out into the cytoplasm. There, a protein-making machine called a ribosome can read messenger RNA to make a particular protein. Every 3 bases of the messenger RNA molecule codes for an amino acid. Proteins are made of amino acids. tRNA molecules help translate the language of DNA and RNA into the language of proteins. tRNA molecules bring the right amino acids that the ribosome links together to make a protein. Proteins are the laborers. Some form structures like tendons and hair. Others perceive light, scents, and flavors, control chemical reactions, and carry messages between cells. To understand our molecular selves, scientists have read the 3 billion letters making up the DNA in the human genome. Different sets of genes interacting with complex environmental factors influence things like our looks, personalities, and risks for diseases like cancer and heart disease. A growing understanding of our genes and all they do will help us understand the complexity and the wonder of life. A simple friendship, a sunny day, a life.
Well, the human genome was completed ahead of time. In 2003, it was published in the scientific journals. And what did we learn? We learned 600,000 slides like this. All right? A lot of letters next to each other. So what these letters mean? Some of these letters we knew from before. I mean, there was research in genetics that dates back, okay, in the 50s and 60s and 70s primarily, but the majority we have no idea. It was a sequence like that, a lot of information. How much information, to make it funny, is this little video from the National Health Services in England, okay? That's the genome length. So saying a letter every second, it will take 100 years. So it's vast, the information is vast. And this is important because, and it changes, it mutates, like in all organisms, all biology, mutation is the rule. So in a way, we'll always be a little bit behind biology. There is a lot of complexity there. So I didn't pick up these 5,000 letters by chance. This is a gene that we knew before the Human Genome Project. It's the beta globin gene. It's a gene that's studied inside out from a lot of years, and is it makes one of the proteins of the hemoglobin, which the hemoglobin, as you know, is the one that transports the oxygen to our tissues. Very important molecule that's been there in evolution from very early, and is very important in humans and all living organisms that breathe. So this particular gene here is not, every letter is not making protein. And this is also important to understand that there are only sections of this gene that are important in making the protein. These are the exons, or the coding sections of the gene, and the black is the introns or intragenic carriers, non-coding sections. And this is something to remember. I will refer back and forth, because I don't want to use scientific terminology, the red letters and the black letters, to remember from this slide, okay? So there is machinery in the cell that recognizes which letters are the important for the protein and cuts them out and makes the important coding for the protein. So there is a lot of mechanisms that can go wrong. That's why every genetic disease has a lot of different ways of abnormality in a way. There is abnormality in the sequence of the letters. There is abnormality in where these segments are cut. There are deletions and duplications of big segments of information. And believe it or not, all of us, we have a little bit of changes like this. So, for example, if in this room, in this section here, I'm sure all of us will differ in some of these letters in the black part. And we think that this is what makes us unique. But in the red...
Dr. Pappas, can I just ask?
Sure.
I never have questions in the middle, but I think it's so relevant.
Yeah.
These non-coding areas, is it that we don't know what they do, or are they sitting there not doing anything?
Oh, well, this is what is happening nowadays. Most of the research happens not so much, it still happens in the red, but the black is what all the researchers try to figure out, because there are elements inside there that have to do with how much protein the cell is making, and how some cells make the protein and some cells don't, because the cells differentiate. You know, the eye cells, for example, they don't make any beta globin. The red blood cells make a lot of beta globin, okay, the precursors at least of the red blood cells. So probably there is coding inside there, and we know that there is for some instances, but not the whole story, that tells the cells how to differentiate, some of them to become sperm cells and others to become neurons and heart cells, and what proteins are different. So, but to come back to the essentials, this little change of this letter here from A to T or C to T, I think it's C to T, I cannot see it from here, from C to T, it's it's the change that's associated with sickle cell anemia. So one letter, okay, makes an individual being a carrier, and if two carriers give this gene to a child, so both of copies have actually this letter change, the child has sickle cell anemia, has painful crisis, has anemia predisposing to infection, strokes. And nowadays, after so many years of knowing of this little letter, finally we have some good treatments about sickle cell anemia. So, coding DNA and non-coding DNA. Coding DNA, most of us, we have exactly the same. And there are changes that actually cause disease in the coding part. In the non-coding part, which is the vast 98% of our genome, the differences are particularly individual, and nowadays we found some changes associated with disease, not actually directly causative. And I'll speak a little more into that because there is a whole science, bioinformatics, people who apply computers and IT technology, information technology, to decipher the elements of the human genome. And it's a growing field and a very much needed field nowadays. So all the non-medical testing that is in advertisements on the TV, and you can buy a little kit and you can do it at home, it uses actually mostly information from the black letters, the non-coded letters, okay, that are unique. This is how they can trace individuals or predisposition to conditions without actually direct link. And in the medical field and the biology, these genome-wide association studies, you may hear it you may see it in newspapers every now and then. What they do, they look at changes in these letters that can be associated with disease without any bias if there's any gene around associated with disease or not. For example, they look at people that have diabetes, and they have this particular letter more than people who don't have diabetes. So they say if you have the little t over there in that particular nucleotide, in association with many other changes in the genome, they can give you a risk. Now, this is not what I do, and this is not what we do in the genetics clinics nowadays. This is predisposition testing that may be something that will be done in the future in the clinic. Now it's done primarily to try to understand common conditions in the research lab, not in the clinics. What we do in the clinics, we do genetic testing. We look primarily at the red letters, okay, the coding sequences and everything that's around them. And how we do the testing is by comparing the patient's DNA with a reference DNA. So the reference DNA was the first sequenced person from the Human Genome Project that is every now and then made it a little better by the more information added from all these people that are sequenced, and a lot of databases. There is the exact databases by the Broad Institute that has close to 60,000 people now totally sequenced, and we have a lot of information that we can use for reference. And we have references with mutations that are specific to disease, and there are many databases of that sort. So when we look at a specific gene, it is a single gene testing or a panel gene testing, when we look genes that are associated with a particular condition, or a genomic test is a test that looks at all our genome. And nowadays in pediatrics, we do in kids that have unusual conditions that we cannot diagnose clinically, we look at the exome. The exome is all the red letters in our genome, okay? The exomes, to try to figure out what's going on. I'll give you some names of the tests just for information. you may have heard, is by looking at the chromosomes, we're looking at these packages of genes, okay, that we can see them by the microscope. And then we can do sequencing by looking at the coding of the gene, and there are in between other tests that look for deleted part or duplicated parts or different process of the genes, which I'm not going to speak today. It's another, epigenetics is another field that we'll talk another time in another instance. And sometimes we go to the proteins and we test the proteins to infer how the genes are there, do biochemical testing. So this is how a chromosome analysis looks. Now these beautiful arrayed chromosomes here are not this how it comes in your microscope if you look at a blood smear of white blood cells that are grown in the lab. They're usually tangled in the top of each other, but we have since 1930s actually by Chio and Levant that invented that particular technique of staining the chromosomes, and by the size and the particular stain, and we know how they're together. So this is pair number one paternal, one maternal, pair number two, and so on. And this is an X and a Y for the male, XX, two X's for female, if you remember from school. This is a karyotype of an individual with Down syndrome. Has 3 chromosomes 21. A whole chromosome 21 is the cause of Down syndrome. Now there are thousands of genes in this chromosome, and you can say why this is not more than Down syndrome. Not all our genes matter if we have them in 2 copies or 1 copy. Very few matter, and actually the genes that matter are the genes that have to do with the neurological development. They matter exactly to have them in 2 copies, one from mother and from father. The genes that have to do, let's say, with the bone development doesn't matter. We have 2 copies or 3 copies or 5 copies. This is another karyotype of individual with Down syndrome with the chromosome 21, instead of being independent, here is on top of another chromosome. Now, we're beyond the 1930s and the karyotype of Chio and Levant. We have a DNA test that arrays 2.8 million little pieces of DNA, and we can see in the lab which is more, which less. So we can see tiny, tiny little piece of chromosomes, and we can see microdeletions and microduplications. Actually, about 15 to 20% of kids with autism have a microdeletion or microduplication that's not inherited from the parents, a new genetic change. Okay? And the DNA sequencers. Now, these are machines that you squirt the blood, they extract the DNA, they do this nice labeling with different colors, and the computer scans, because everything is so microscopic at the level of integrated circuits on a slide, and they get the sequences very, very quickly, and you can sequence a genome like in less than a day now, by all with no interference by pipetting or human interference. Everything comes automatically. So what do you get from the genetic testing? What's the result? You can get the pathogenic variant. And now in the testing that we do in the red letters, okay, pathogenic variant means like the one for sickle cell, a little change in the letter that causes disease. A likely pathogenic, which means that we predict with all these models from bioinformatics that we have in the computer, that this little variation is going to destroy the protein or the cell or whatever, but we don't have a lot of clinical information. We don't have a lot of patients there. That's likely. And we can have variants of uncertain significance, which means that we have no information. It's nowhere in the databases. We find something that's different than the reference, but we have no information. And this is one of the limitations, the drawbacks, and the dilemmas that can start, that you can see some started creating by genetic testing. So that's why informed consent is there, to give this information and all this information that is in the slide, including information about insurance. People ask, my medical premium is going to go up, okay? If I do this genetic test and they found that I'm predisposed to have breast cancer or whatever. Now, 2008, Genetic Information Non-Discrimination Act is a federal law that prohibits raising premiums for medical insurance, prohibits discrimination in the workplace due to genetic results or genetic diagnosis. However, the life insurance is out of this. So life insurance, they can adjust premiums according to whatever test you're doing. The other thing that we discuss is paternity. I mean, once we do family studies, you may find something that's unexpected or not discussed, so we have to discuss it. So let's go to a couple of cases. This is a very bright young guy, a college student who actually was an excellent swimmer, and he was doing long distance swimming for fundraising. And he did it quite a few times, and one time after his dive, he had a cardiac arrest. But now a few months later, I see his parents. It was very early actually in my, it happened here in NYU. The parents came and with the three siblings, and they say we went to cardiologists, all of us, we had EKGs, our rhythm looks fine, but they sent us to genetics. So what we know about the genetics of cardiac arrest is that there are a lot of different conditions, but one group of conditions is common. It's called long QT syndrome. It has to do with the electric stimulation of the heart and how it's going around, and it has to do with the genes that regulate the salts that have to do with this electrical stimulation. So there is, in particular, actually, a gene that's called KCNQ1 it has to do with cold water and diving in cold water and cardiac arrest. So we knew where to look. We didn't have to look in all these letters, We knew exactly where to go to look for this family. And sure enough, the father and two of the siblings had a genetic change. Now, this can be tricky. Why it can be tricky? Because approximately 20% of the families that meet the clinical criteria, this family didn't even meet the clinical criteria, that meet the clinical criteria for long QT, we don't find a mutation because we don't know all these genes. We know, as I told you, 1/3 of them or so. So in this family, we were able to find it, but we can do the genetic testing. We can do all the genes that we know that are associated with long QT, which is about, it's more than 15 nowadays. We don't find anything. This is false reassurance in a way, because there will be other genes there that we don't know that are particular to this family. So this is important from the genetic counseling point of view to say the genetic test is good if we find something. If we don't find anything, it's not reassuring. It may still be there. But if we find something, it's important because there are medications for this. Now, for other genes, you have to use defibrillators, but for this particular gene, you give beta blockers, you give medication, and you decrease a lot the risk for heart attack, all right, for cardiac arrest. So, and it's important also for the rest of the family members, and that you open a kind of worms actually, because you cannot, because of HIPAA regulations, you cannot communicate these results with other family members. You have to convince these parents to go to their siblings and so on, and go for their evaluations. But you can save lives. This is another story. This very athletic, actually 38-year-old, was going to the meeting with the obstetrician of his second child. Now, to orient you a little bit about the family histories, the squares are males, the circles are females, and the little rhomboid here is an unborn child. So he was running to catch the appointment, the obstetric appointment, when his wife was pregnant, and falls down and has again a cardiac event. Both him and his father were sports lovers, but his father, as we see here, died early, age 45. This line means death at age 45. And they thought that it was cardiac, but there was no autopsy done. They didn't know, I think, in this case. So, just for interest, it happens in this family is a dissection, as we call it. Dissection means that one of the layers of the aorta, which is the big vessel that comes out of the heart, it breaks and blood goes in between and obstructs the circulation. And this dissection has to do with a propensity for one of these layers to have problems. So what's there? It's connective tissue, the tissue that's gluing everything together. It's also muscle fibers, smooth muscles that are important in the dilatation and the aorta movements. So this particular individual had an abnormality in one of the smooth muscle actually genes by doing a lot of genes here, and some of them are connective tissue, some of them are muscle. It was in the smooth muscle gene, and what was important is that his first boy, that was 2 years old or so, had a little vessel that was open, that usually closes by itself after birth, that had to have an operation to ligate it. It's called the ductus arteriosus. And this is something that goes along with this particular gene. So we knew that his son also had this gene just by clinics, just by clinical evaluation, but of course we did the testing, and they wanted to do prenatal testing also for the baby that was ready to be born, and we did it. They didn't want to do it for continuation or termination of the pregnancy. They wanted to be ready in case this vessel was still open to do the operation much earlier than the son, and that little baby also had the little change there. This is autosomal dominant predisposition actually to disease, 50-50. It's 50% in every pregnancy. And there is no memory. It's like flipping a coin with every pregnancy. And how this evaluation helped this family, I mean, this is obvious. And for this dissection and preventing other dissections, there are things to be done. In this case, we tested the babies, but we don't test kids for adult consent conditions. And this is a consensus that we came to the American College of Medical Genetics after a lot of deliberation, because we don't want, in a way, to stigmatize a child as growing on for a condition that is going to show at age 30 or 50, or even later when you think about Alzheimer's and other conditions. This is a pediatric case. This is a baby that had actually a dissection in the emergency room, and nobody thinks about a dissection in babies, and then the pathologist after the autopsy comes and says, look, I have a little baby with a dissection, what is this? And if you look in the family history, there is a history of dissections here in this family that was never taken seriously, I guess. And some of these conditions that are adult onset, some of them, you can see it also in pediatrics. And one of them is this type of dissections. Now, before I go to the cancer, I have to tell you that here in NYU, there is a great arrhythmia center. You remember the swimmer and the arrhythmia problem by Dr. Cerrone, who takes care of families with this genetic predisposition to cardiac arrest. And this is a great resource. At the end, actually, of this talk, I have a slide with all the genetic resources here in NYU. But I wanted to mention that since we're going to move away from the heart a little bit. Now, in cancer, genetic testing took a lot of dimensions, but it started with testing like this. For example, this individual here says, my grandma, my paternal grandma, died from ovarian cancer. Am I at risk or not? And she was very astute because the gene, of course, can go through her father. Her father has no ovaries, you know. Although we know that these genes nowadays can be associated with prostate cancer and pancreatic cancer too, but not so high as it is for ovarian and breast cancer. And sure enough, again, we found a BRCA1 mutation in her, and it helped very much her aunts. One of them had also the same mutation. Now, her father did not want to know anything about all that. He said, don't involve me. I don't want to know if I have genes or I don't have genes. I don't know. You know, this is more associated with the Y chromosome. I think, well, we men don't like doctors so much. So, but anyhow, he had to know, because if his mother and one of his sister and his daughter were carriers of that, he's an obligate carrier, as we say in genetics. So in a way, it was imposed on him. It was imposed on him to have a little more of his prostate stuff and to think about the pancreatic that he didn't want to even. So this is again an ethical thing in families, and families in a way have to come to a consensus before you take the lead of this type of matters, you know, before you go to the genetic counselor. Now, testing in cancer is not this germline testing. This germline is the test that you see the genes of every cell that we have. But there is tumor testing also. And tumors have their own genetics. They don't have your body genetics. They develop their own mutations as they go on. And these mutations are very important because there are specific therapies. What is thought as personalized medicine is probably, this is the best example. Personalized medicine means look at the tumor and give the exact indication. For example, tumors that have the BRCA1 and BRCA2 mutations are very susceptible to PARP inhibitors, a group of medications that deal very well with breast and ovarian cancer, and sometimes pancreatic too. All right? Yes, the oncotype is this one. It's this one. The oncotype doesn't tell you what all the cells of your body have. It tells you what the tumor has. So the genetic clinic reality come back to my reality, because I belong to the Department of Pediatrics, and for many, many years we were doing everything. We were doing the heart genetics, the cancer genetics, the whole thing. Now we have here in NYU a very good cancer genetics program with 8 genetic counselors and Dr. Kingsburg. There's a prenatal counselor that does all the prenatal stuff. And we have Dr. Heather Lau, who does enzyme replacement and gene therapy for severe pediatric disorders. So things are happening here too. This is to tell you what we are, what are the genetic professionals. Clinical genetics, my specialty is a primary specialty now. It's one of the 28 primary specialties, like pediatrics, OBGYN, and internal medicine. So it has its own program and its own board certification. And we work with the genetic counselors, which are masters they have a master's in genetic counseling, and we work with laboratory geneticists that are PhDs and MDs. Cytogeneticists are the ones who deal with the chromosomes, moleculars with the sequence, biochemicals with the proteins that are abnormal because of genetic disorders. So this is the what we're dealing in our clinic with diagnosis primarily, and we're involved very frequently in cases in the hospital that are very complex and the diagnosis is not obvious, and infectious disease, and the rheumatologists and everybody else have given their consultation, and then what's left, let's call the geneticist. Okay, so we frequently part of a team with a patient that's difficult to diagnose, but we're involved also with carrier screening, and this is important. We see a lot of couples nowadays that they are tested for autosomal recessive conditions. Okay, we have a panel of genes that are approximately 300, 280 something that we test for recessive conditions. So if a couple are carriers of the same condition, then they have the option of doing even pre-implantation genetic diagnosis, okay, before they implant the embryo for this type of conditions, and this is very popular. And of course the cancer and prenatal stuff. These are two slides, and this slide and the next one is from Elsa Rich, who is one of the first genetic counselors here in the US, and has put together of questions that the parents actually ask us after we make a genetic diagnosis. And it's very difficult actually to say to a parent of a child that has a genetic syndrome that this has nothing to do with whatever you did in your life. You have no say which actually genes you pass down, and you have no say into creating a new genetic change, a new mutation. These are all biological phenomena, and there is no exposures or anything that can change your DNA. Is very well protected, short of exposure to a nuclear explosion or a nuclear accident. The DNA is not going to change. It changes only from the gamma rays that come from the cosmic gamma rays that come from the outer space. That's the only thing that we know that changes our DNA. All right, it's very well protected. So all these questions there that you see, and make sure that the parent's guilty when they have a child or there is somebody in the family with a genetic disorder. It's all biology, and biology has no feelings. It happens. So what's the future directions? So the future directions it's actually the first one, genomic tests that will look at all the genes and also these black letters, all this DNA in between, and try to get an idea of the severity of genetic disorders or non-genetic disorders, because genes have to do with predisposition to infection, diabetes, thromboembolic episodes, or strokes, or heart diseases, that they have a lot of environmental component, but they have also a genetic component. So all this testing, it's going to end up give us an idea of how to manage and how to diagnose the common condition. And precision medicine is exactly that you look at the genome of an individual and you can give the medications that are more likely to work, or the treatment. There is a study that was initiated by the NIH that the target is to enroll 1 million people here in the US, and it's happening very fast, and they record all the medical information plus all the genome information from these particular people, try to make sense and try to decipher how to associate disease, genetics, and environment. The newborn screening in the future, which is now limited to about 80 conditions that are treatable, and it's done in every newborn that's born in the US, by a little blood on the hill. In the future, it may actually be a genomic test. And people start piloting that, doing the pilot studies for this to become a reality. So, conclusions. Is the genetics, is knowing your genetics right? If it's actionable, like the cardiac and the cancer cases that I showed you, of course. Sometimes we do this genome testing or exome testing on kids, and as they look at all the genes, they find incidental actionable genetic findings. For example, they find a mutation in BRCA1 or BRCA2, and we give this option to the parents to know this information. So some parents actually take this option, the majority of the people do. Sometimes even if it's not actionable, genetic testing can provide a diagnosis closure. Like in a very severe condition that nobody has an idea how this came about, if you have the diagnosis, it may be important. It may inform reproductive decisions, and as you say, with many research studies, actually, it may help developing new medications. And another fun fact is that we share a lot of genetics with other species, including the bananas. All right. So 60% of our genome is common with the bananas. The idea here is that the majority of our genome is basic biology. It's basic biology of the cell that is there. And actually, tweaks in this basic biology has severe consequences. That's why pediatrics was the first specialty to deal with this type of disorders, because the kids were born with very severe problems. And this is how we started looking at the genetics. And don't forget family history. I look like I belong here, right? But if you look at your family history and looks like a duck and walks like a duck, okay? If you look at, and half of your family history has a double chin like me and the belly and diabetes, and you have the same double chin and the belly, you're most likely going to develop diabetes, right? You don't have to do expensive genetic testing for that. You can just look at your relatives. This is what I promised you to show you how much genetics is nowadays, clinical genetics, not research. Research is much more. But all these people in our center are involved into clinical genetics. I would like to pick Dr. Marina Cerone for the Inherited Arrhythmia Center here in NYU. The Dr. Heather Lau, who does the treatments for genetic disorders, and the big cancer genetics program. Thank you very much.
Thank you, Dr. Pappas. I invite you to sit on this chair. Turn the lights up. So that was great, and you can see how it's such a huge topic. It's so complex. There's so many questions. I'm going to throw two quick questions at you, and then we'll take your questions for another 10 minutes. And one is, are we able today to do anything about a genetic sequence where we splice out the bad one, put in a new one? Is that reality, or is that coming down the pipe? That's my first question.
It is reality. It is reality. Actually, the very first FDA-approved gene therapy is for spinal muscular atrophy. The very first child that was treated was here in New York. It was a little girl from Harlem. That was 3.5 years ago. So what this genetic therapy does is that it has a mechanism that wakes up a gene that is usually not working and helps the gene that's mutated. Spinal muscular atrophy kids, they don't move after they're born. And eventually they die if you don't put an endotracheal tube to help the breathing, So you have to have them with a trach. So this little girl that got the gene therapy is running around.
Wow.
Is running around. It's like, it's amazing. So this, I'm not saying that gene therapy will be like that for every condition, because a lot of genes, because of their basic cellular functions, they start working from the time that we're conceived, when we were on one cell. So this cannot be reversed going back there. But in this case it works. In some other cases it will work.
So
also.
what practically are they doing? Are you taking out the bad gene by doing
Actually, you wake up a gene that's dormant. That is dormant by evolution. We have a lot of duplicated genes in our genome that they silence, another gene takes over through evolution. But this is how they do it for SMA. But all these CRISPR techniques that you read in the papers, they can be done. Clearly, they can be done in vivo and ex vivo, meaning they can be done by an injection in your blood or in the cerebrospinal fluid, the brain, or in the liver or whatever, or they can take the bone marrow out and they treat it and they can put it back. All this type of therapies are some of them are in very late stages in
It's
the
very
development.
exciting. Which brings me to the second question. You touched on it, but the whole field of epigenetics, which maybe we'll have you come back and talk about that. And my understanding is that you're born with certain genes, but really you have a huge influence as to whether you can express these genes or not, good ones and bad ones. And I'm very big on that, talking to my patients, because you can be born with genes that put you at risk from diabetes, that put you at risk from heart disease, that put you at risk from arrhythmia. But your lifestyle and how you live can express these genes or not express these genes. In other words, your genes are not your destiny. I'd love you just to comment about that. And this is an emerging field. And what you feel, we can do, these people, we're sitting here listening to you, do we have any benefit of actually knowing some of our genes? And that was one of my questions to you. So we can do certain lifestyle changes, or do we just go, look, just do all the things we're supposed to do and hope that you're not going to express a bad gene that you may have?
So clearly, epigenetics is there. I mean, there's a whole field of how environment and gene interacts. But the more we learn about epigenetics, is that the more we learn that there are other genes that influence other genes in our body. For example, most of epigenetics happens in the nucleus of our. But environment plays a role. There's no doubt about that. But from studies, as you see, I don't want to be doom and gloom, but from population studies, you see, there are a lot of people that are predisposed to diabetes with the double chin and my belly, right? And you can try to coax them to exercise and diet. It doesn't go so well.
No, but
So it seems that there is a lot of program there.
If they do it, that is a problem. But if they do it,
If they don't do it, yeah.
you can land up not expressing a bad gene. Any other questions? Yes.
I'm not sure I used the right terminology, but I think you mentioned autism.
Yes, I did.
And there are so many children now being diagnosed with autism. Is there a way to check genetically?
Sure.
So let me repeat it because this is being recorded. The question is about autism. Is that something predictable from the genes?
Sure. So there are two reasons that the frequency that we diagnose autism actually increases. One is the diagnosis, and this is proven over and over again, that the diagnosis of autism has educational benefits. So people that have kids with intellectual disability, they prefer the diagnosis of autism than intellectual disability. And this is clear. It's proven over and over again. The other part is that it may be a real environmental factor there that wakes up the genetic predisposition. The genetic predisposition was there for ages, but now we see it. Why we see it, probably there are environmental factors there. But the genetic part of this duo, environment and genes, is testable. So we know that the microdeletions and microduplications that I mentioned before, and we know about 350 genes that are associated with autism. There is the Sfari and Simons Consortium that collects families and for autism, and clearly we have huge knowledge about the genetics and how and the architecture actually of the brain of autism is coming together. Soon we'll have medications, but the medications will be it's important actually to make the diagnosis correctly in order to apply the medications correctly.
So you can actually test. Is that something that you can actually do in the internet and tell a parent? I mean, it's not, or because I think that's what you're getting at. I mean, it's very, very rough information to have to tell a parent, but if you do know that, then clearly there are interventions that you can start very early on. Are we at a point where you can, if someone says, is my child likely to be autistic, you can give them that information?
Sure. So the pre-symptomatic diagnosis in genetics is a chapter. So pre-symptomatic, it doesn't go only for autism, it goes for adult neurodegenerative diseases, Alzheimer's, Huntington's disease. Good people like to know pre-symptomatically what's going to happen to them. So there are centers, and there's a center in New York that they have the psychological support for adult neurodegenerative conditions. For kids, we don't do pre-symptomatic testing. We decided, as the American College of Medical Genetics, we don't do pre-symptomatic testing. We test only people, kids that have the condition.
Okay.
It's not so much to confirm, but to give tools and information of how to manage the child. The diagnosis will be there by the psychological tools that we have, ADOS, ADI, all these scales that the psychologist has to make the diagnosis of autism. But if you know the gene, then you go to other cases that have the same genetic change and you learn. You learn about the future, you learn what's the best management, behavioral versus medication versus others. For the time being.
So I know for breast cancer, you have to provide ethnic
Information.
background. Is it the same for other? Do you repeat that?
So the question was, when you have breast cancer screening, you provide ethnic information and other information. The question is that the same for all testing when you're looking at cancer genes?
So, when Dr. Mary Claire King did the initial studies and found the genes for breast cancer, found a genetic change initially and 3 at the end that were more common in Ashkenazi people, and made the test back then, before the Human Genome Project, feasible because it was only for 3 specific letters or letter combinations. Nowadays, that we sequence the gene, the ethnicity doesn't matter so much because we no matter where you come from, we look at the whole gene. And even in people of Ashkenazi origin, if we don't find one of these 3, we continue to do the rest of the gene. And in many people of Ashkenazi, we find other mutations. And it's been reported also that the Ashkenazi mutations are people that they don't think they're Ashkenazi, but probably are. So it doesn't matter. It doesn't matter nowadays because the technology does not rely on specific genetic changes. We look at all the genes.
So what do you think about these companies out there, 23andMe? There are many of them. They're supplying a lot of information, certainly about your family. You can find out you've got an half sister and half brother somewhere. But what about medical information? Do you think there's utility? Do you think that we're opening up a bag of worms and it's becoming very popular and there are many companies out there?
So if they have, for example, 23andMe has embedded in the test the 3 Ashkenazi Jewish mutation for breast cancer, they're there. But if there is a hit, I suggest to confirm it in a clinical genetic service or with your oncologist, whatever, because we had the way they do the test with genotyping is an old technique. Now, they will catch up, and the technique is going to get better, and all these direct-to-consumer testing will be there. Nobody will take it out or outlaw it. I mean, we're a free society. Things will be there. The bioinformatics tool to tell you what is what, it's expensive. The expensive part of the genetic test nowadays is not do the sequencing or reading the biology. It's actually decipher what it means. Do the work, the real work of finding out what actually the meaning of all this stuff, and looking also at the clinical part of how the individual looks. So these companies do not have this. So for a good genetic evaluation, I think when it comes to be important for your care, not just as a gimmick, you have to go to a medical center.
So there are companies out there that have bioinformational systems built in, because that's the key. They do. You can analyze 5,000 SNPs. You don't want a book with all the SNPs. You don't know what's going on. So they give you a report of these are the actionable SNPs. So they filter it down for you, and then the best thing is to take it to someone who can help you, because otherwise it just creates tremendous anxiety. Because as you pointed out, just because you've got a gene, it doesn't mean that it's going to express itself. And so this is a huge field where it's very, very important to be talking to the right people so that you don't get over anxious about nothing, and at the same time not ignore something that you can really do something.
To add to that, at this point, we're at the stage of dealing with the clearly pathogenic variants. This predisposition testing of risk assigning, we don't do it in the clinics. Some clinics try to do. There is a clinic, an adult clinic at Baylor in the human genetics department of Baylor who gives you risk for cardiovascular disease, but it's not well curated yet, I think, our genome for this type of use. So we need a little more work for that.
Thank you so much. I really appreciate it. Thank you all for coming in this weather. Really appreciate it, and be in touch with us and we'll let you know what's happening in the next few months.